Good morning, AP biologists. We've been talking a lot lately about the structural components of the cell membrane and how those structural components contribute to all of the different functions our cell membrane has. We've talked a little bit about the semi-permeable barrier that lets some stuff in while other things cannot pass through the membrane. And we've talked about how those channel proteins can help transport materials into and out of the cell that can't easily pass through the phospholipid bilayer. Today what we want to do is talk a little bit about how materials move across the membrane, how that actually leads to dynamic homeostasis. So, first thing we need to understand is that particles are in constant motion, and this random constant motion is going to cause particles to naturally spread out throughout their motion. Now, if we separate these particles by a membrane, the process still holds true. So here we've got a large concentration of these particles inside this membrane, and based on their constant random motion, they're going to spread out. So we're going to have particles on the inside and the outside of the membrane. Now this is really, really important when we talk about the diffusion of gases in our circulatory system. So our blood cells are going to carry a high concentration of oxygen. That oxygen will move to our cells, which have a lower concentration of oxygen. Our cells are going to have a high concentration of carbon dioxide. Our blood cells will have a low concentration. So the carbon dioxide will move into our blood cells so that it can be brought to our lungs and we can breathe it out. This type of transport is called passive transport because it does not require any energy. And the particles are going to move from an area of high concentration to an area of low concentration. Again, both of these things is because these particles their motion is based on the random movement and constant movement of these particles. So they're going to constantly be moving and naturally spread out. The overall result is that equilibrium is established. We have an equal concentration inside versus outside the cell at the end of this type of passive transport. Now, let's talk a little bit about diffusion. This is one of the types of passive transport. This is going to be the movement of particles directly through the membrane from an area of high to low concentration. So you can see on the left, we have that high concentration. On the right, there's low concentration. These particles are going to diffuse right through the membrane, and we're going to have equal concentrations. Now, let's think a little bit about what type of molecules are going to be able to diffuse directly through the membrane. Exactly, small non-polar molecules are going to be able to fit directly between the phospholipids and be able to diffuse through that hydrophobic tail part of the membrane. Again, let's take a closer look at our red blood cells. High concentration of oxygen inside of our cells, blood cells, a low concentration of oxygen inside of our body cells, and so these cells are going to diffuse to even out the concentrations, and now we have delivered oxygen to our cells in our body. Now, sometimes we have protein channels embedded in the membrane, and this is going to help with facilitated diffusion. This is where the particles are going to move through the channels, again from high to low concentration. Let's think about what type of molecules are going to need protein channels in order to diffuse. Exactly, these particles are going to be slightly larger, so they can't quite fit directly between the phospholipid in the bilayer, or they're going to be polar, and so they have a charge on them or an area of a positive and negative charge, and they can't actually fit, they get bounced out by that hydrophobic region of the phospholipid bilayer. Let's take a closer look at the inner membrane of a mitochondria to see how we might use these protein channels. Our mitochondria create this high concentration of hydrogen ions on one side of the membrane and a lower concentration on the other, and these particles are going to diffuse using the protein channels embedded in that inner membrane to create equilibrium, and this process is actually going to help us to drive the production of ATP. Now, let's take a look at this scenario. We have a high concentration of particles on the left and a low concentration of particles on the right, but the particles are too big to fit through the channel or the membrane. So what do we do now? Well, don't forget that solutions have two parts. Not only do we have the solutes, the particles, the ions, the sugars, the whatever it might be dissolved in the liquid, but we have the liquid. In this case, in our bodies or in living things, it's water. So instead of the particles moving, the water can move to create equilibrium. So let's talk a little bit about osmosis. Now, If the concentration of two solutions is equal, they're considered isotonic. So inside the cell and outside the cell have the same number of particles, therefore these solutions are isotonic. Water is going to move to the right, water is going to move to the left in the same amount, so we really have no net movement of water. Now, let's talk about when the solutions are different. So the first thing we need to do is define our solution. So some solutions are hypertonic. They would have more particles, less water. They are more concentrated compared to others, which would be hypotonic. They have less particles, more water. They're less concentrated, or we might say more dilute. Now remember, guys, these are terms that go hand in hand. They're comparative terms. So on the left side, we have a hypertonic solution compared to the right side, which is hypotonic. Now, when we have solutions that have a differing concentration, water is going to move. So let's take a look. On the left, our solution is hypertonic, more concentrated than our solution on the right, which is hypotonic. Water will move in both directions, but more water is going to move towards the hypertonic solution in order to dilute it and even out the concentrations. So the result of a scenario where we have a hypertonic solution and a hypotonic solution is that water is always going to move towards that hypertonic solution. Again, the movement of water is just based on that random motion. Now, let's take a closer look. Let's zoom in so we can actually see what the particles are doing. Notice on the left-hand side, I have a higher concentration, so it is hypertonic than the right side, which is hypotonic. Now, we can watch the movement of water towards the hypertonic solution. As the particles move, we can see how the concentrations change. The concentration on the left-hand side gets less concentrated, and the solution on the right-hand side gets more concentrated, until eventually our solutions have evened out, and now our solutions are isotonic, and we no longer need any net movement of water. So remember, water is always going to move to create equal concentrations or equilibrium. Now, this becomes really, really important in living systems. So here's a red blood cell in a hypertonic solution. We know that water will move out of the red blood cell, but this can be really disastrous because our cells can shrivel up and then they no longer function. But the opposite is equally disastrous. If we place our red blood cells in a hypotonic solution, the cell itself is hypertonic. The water is going to rush in and our cells are going to expand until they could possibly burst. We call that lysing. Again, not good. Let's take a look at plants. Plants are a little different. When plants are in hypotonic solutions, the water is going to rush in, and this is actually a good thing. It's going to create turgor pressure, or we say that the cells are turgid. And this is good because cells are going to need that turgor pressure in order to remain rigid and have the structure of the plant itself. Now, when they're in an isotonic solution, water will move in and out at equal rates, but we say that this cell is flaccid. It starts to wilt a little bit because it doesn't have as high of a turgor pressure. Now, if we have a hypertonic solution, water is going to leave the cell, and this is really disastrous for plant cells because the cell membrane actually pulls away from the cell wall, and we say that the cell has plasmolized, and this is fatal to plant cells if they're in hypertonic solutions and they lose too much water. So this idea of osmosis is really, really important. Now, remember, all of these types of passive transport are based on that random, constant motion of our particles, which tend to move our particles away from each other and spread them out. So this is how we can create equilibrium inside and outside of our cells without expending any energy. But there are some times when we want to create a high concentration of a particle on one side of a membrane. So we're actually moving against this constant, random, natural process of the movement of particles. So this is going to require energy. So let's take a closer look at our inner mitochondrial membrane one more time. So we have these protein channels embedded in the membrane, and they are going to actually use ATP energy to pump hydrogen ions out. So we create this really high concentration of hydrogen ions outside the membrane and a low concentration inside the membrane. This is going to allow this high gradient to be formed, which then these particles can rush back in, and we can use the release of energy from the passive transport to fuel our production of energy by our mitochondria. But in order to create that high concentration, we need to add energy first. So this is considered active transport. So here on the left we have high concentration, on the right we have low concentration. Now normally these particles would randomly move to diffuse to create equal concentrations, but we're going to instead make our high concentration even higher, our low concentration even lower. In order to do that, we have to expend energy. We also have bulk transport, where we're moving really, really big particles, cells or proteins that the cell has created, or food particles, anything like that. So exocytosis is where we're going to have the internal vessels fuse with the membrane, and they're going to secrete large macromolecules out of the cell. So maybe the cell is getting rid of some large waste products and expelling this little particle, or maybe it's produced a big protein that it needs to secrete out. Now, the opposite is called endocytosis. This is when the cell is going to take in macromolecules or other particulates by forming a new vesicle from the plasma membrane as it changes shape and brings the particle in. So this is all really, really important. So those are all the types of both passive and active transport. They help a cell maintain and create homeostasis. Thanks for watching.